Dear organizers of the conference and its participants, I want to introduce you the main results of our research named CKIM's participation in hormonal and proliferative activity of normal endometrium and endometrial carcinomas. Let me present them in the following structure. At first, topicality, introduction, aims of the study, then material and method, results, and finally conclusions. It's well known that endometrial cancer is the most common cancer of the female genital tract. Approximately 3% of women will develop it in their life. The incidence of uterine cancer has been increasing in recent years, and despite the great achievements in treatment and diagnosis, the risk of mortality from this pathology remains high. In addition, late diagnosis may lead to worse survival. This led us to search for new molecular markers that can help in early diagnosis and in understanding the malignant processes in general. One of those indicators of tumor progression may be carcinoembryonic antigen-related cell adhesion molecules, also known as CKIMs. CKIMs are members of the immunoglobulin superfamily. There are more than 17 genes that belong to this family, but the most studied are CKIM1, 5, and 6. They take part in many functions, such as regulation of apoptosis and proliferation, enhancing the inflammatory response. They also stimulate the differentiation and polarization of epithelial cells, and take part in ducts and glands formation and in many other functions. CKIMs were found in the numerous cell types such as endothelial and hematopoietic cells, as well as epithelial cells of different organs. Their expression is decreased in numerous tumors, such as carcinomas of breast, colon, and liver. At the same time, their upregulation is observed in lung cancers and pancreas cancer, in melanoma and tumors of ovary. Unfortunately, the data about their participation in tumor progression is limited. Information about their role in hormonal and proliferative activity of endometrial tissue is controversial. Given the above, we decided to solve these numerous misunderstandings in this scientific problem via investigation of CKIM1, 5, and 6 expression in normal and malignant endometrium and their influences on hormonal and proliferative features of endometrial tissues, with following possibility to use them as a reliable tumor marker. The study was conducted on 50 samples of endometrial carcinomas, 30 of which were endometrioid carcinomas and 10 cases of serous and clear cell carcinomas. All tumors were classified according to World Health Organization with the determination of the degree of their differentiation. Normal tissues in the amount of 12 were taken from uterus after hysterectomy. The expression of the previously listed receptors was studied by 3 immunohistochemical methods: immunoenzyme, immunofluorescence, and double immunofluorescence staining. Monoclonal antibodies that you can see on the slide were used in this study. During the double immunofluorescence staining, the main condition was to use both primary antibodies from different species, mouse and rabbit. So I want to present our results. Let's start with the normal endometrium. We found CKIM1 expression on the apical surface of the endometrial cell, regardless of the menstrual cycle. The same thing you can see during immunofluorescence study. Regarding other CKIMs, we didn't find CKIM5 expression in normal endometrial tissues, and CKIM6 was characterized by rare expression sites in endometrial glands. Next, we consider the expression of CKIM1 in endometrioid carcinomas. You can see that apical expression of this protein remains in highly differentiated tumors, but with the loss of the glandular structure, the expression patterns of this protein also changes. It acquires a character of membranous and cytoplasmic. In addition, there are areas that are completely devoid of expression of this protein. For serous and clear cell tumors, all patterns of expression were also characteristic: apical, membranous, cytoplasmic, and lack of expression. As for two other proteins, upper panel demonstrates the focal expression of CEACAM5 and CEACAM6 in endometrioid carcinomas. For type 2 tumors in the bottom row, you can see the diffuse expression of these proteins. I want to remind you that CEACAM5 was not expressed in normal tissues. Then we studied co-expression of CEACAM1 with progesterone and estrogen receptors, at first in normal endometrium. Using serial slices, it was revealed that the steroid-positive endometrium, which you can see in the upper row in the proliferative phase, was characterized by the expression of both hormonal receptors and by expression of CEACAM1. At the same time, the endometrium in the secretory phase, in the bottom row, had weak expression of receptors for hormones, but it retained a high expression of CEACAM1. As for malignancies, endometrial carcinoma had different combinations of these receptors, when both CEACAM1 positive and CEACAM1 negative tumors had as steroid positive as steroid negative variants, phenotypes. So we didn't find the relationship between CEACAM1 and hormonal receptors in normal and tumor endometrial tissues. Our next aim was to investigate the possible effect of CEACAM1 on the proliferative activity of cells in endometrial carcinomas. It's seen that the areas of neoplasias with CEACAM1 positive expression have lower rates of cancer cell division compared with CEACAM1 negative areas. Moreover, the lowest cancer cell division is in the presence of the apical expression of this protein. Finally, inhibiting effect of CEACAM1 on the proliferative activity of neoplastic cells was proved in the double immunofluorescence staining. It can be noted that hot proliferative sites of endometrial carcinoma are CEACAM1 negative. Conversely, cold proliferative sites of endometrial carcinoma mostly express CEACAM1. All mentioned above give us opportunity to make following conclusions. At first, CEACAM1 take an active part in the functioning of the normal endometrium. Neoplastic transformation accompanied by variability in the expression of CEACAM1, 5, and 6 in the number of receptor-positive cells and in expression patterns. Besides, CEACAM1 doesn't affect the sensitivity of endometrial tissue to progesterone and estrogen, but inhibits the proliferative activity of neoplastic cells. And finally, the study of CEACAM1, 5, and 6 expression may be a reliable tumor marker of, as well, emergence and progression of tumors. And in conclusion, I want to draw your attention to the table where we show how the results of our research may possibly used in clinical practice, namely in the diagnosis of endometrial carcinomas. So thank you for attention, and I'm ready to answer your questions.
Okay, thank you for your report. I'd like to ask our judges, our jury members to ask questions if they have. And while they are thinking, I can ask my question to you. In your work, you mentioned that normal endometrial tissue was obtained from the uterus of women operated because of leiomyomas, right?
Yes.
And but can this endometrial tissue consider it to be normal one? Because as we know, there are a lot of changes, possible changes of adhesion molecules expression in condition in which leiomyoma develops. I mean, you know, when hyperestrogenemia or another changes can lead to myoma development. It also can lead to changes in endometrium, and you use this endometrial and consider it as it's normal. Are you sure if it was normal?
Yes, thank you for your question. In our study, we call normal endometrium the endometrium that have not malignant changes. Yes, it can be a possibility that it will have some changes that can change our results, but unfortunately, we don't have a possibility to take intact endometrium.
Okay, thank you for your question. Are there any questions from our judges?
Maybe I. Thank you, very important report, but I have a question. How have you estimated the proliferative activity of endometrial cells in your work?
We have used the Ki67. It's known as a reliable marker of the proliferative activity of cells, and with the usement of this with anti-Ki67 antibodies, we have examined the proliferative activity.
Thank you. Thank you. And please read in the chat. Next question.
In the Viber?
Can you explain the disappearance of CKM1 from the tumor cell's surface?
From the tumor cell surface. Yes, thank you for your question. I can explain this in this way. It can be a manifestation of antigenic simplification. As we know, some tumors stop expressing some proteins during their malignancy changing. So I can explain this as antigenic simplification or antigenic complication.
Yeah, actually, usually we call this like tumor progression and kind of dysplasia, right?
Yes.
Dysplasia. Okay, thank you very much. Are there any questions? Any more questions?
Yes, please, if possible. Good morning to everyone. I'm Vinogradova Elena, Department of Physiology. And Vladimir, thank you for your report. I have few questions to you. First question, can you say, maybe you know some sources of literature, or maybe you know how early maybe possible to use CCAM expression for indication start development of endometrial carcinoma? Is it possible to indication and which stage of cancer?
Yes, we think that it may be possible. Unfortunately, it's not possible nowadays, but in future we hope that it will be possible and it will help in more earlier diagnosis of these tumors.
Mm-hmm. Okay, thank you so much. And I have one more question to you. I want to know, so interesting to me, what is your participation for preparing this huge work? What did you do?
Thank you. I take part in these immunohistochemical methods. We will prepare together and we will prepare together immunohistochemical reagents, but sections, slices, and by me they were also observed, and we have then make conclusions and results.
So your participation more in theoretical part, as I understand, than a practical part. Is it true?
Yes, in practical and theoretical part.
Mm-hmm. Okay, thank you so much.
Okay, and we have another question from the chart. And the question is, what is the normal role of CCAM1 protein in the womb, in the uterus?
Thank you for this question. It's known that CCAMs are proteins that belong to the immunoglobulin superfamily, and it's also known that they have properties against microorganisms. So perhaps they can interact with bacteria and contribute to their clearance and maybe prevent their penetration into the mucous membrane of the uterus. So in other words, CCAMs interact with the environment. For this, they are expressed on the apical surface. It's one of the main functions of these proteins, in our opinion.
Okay, thanks a lot. Are there any more questions? If there is no questions, let's move on to the next contestant.